All right, let's start the 14-2 podcast. And this podcast is all on human chromosomes. And we're going to be looking at a couple of specific situations with that have to do with the X and the Y chromosome and inactivation of the X chromosome and some chromosomal disorders that occur during meiosis. So, Um, the two smallest chromosomes, 21 and 22, were looked at pretty early in when DNA was being researched, and those two chromosomes were, as you can imagine, if they were small, a lot of things were identified on them very quickly. And you can see over here, here's some right here. There's ALS, which is known as Lou Gehrig's disease, and here's a form of leukemia over here that was found on number 22. So they found some pretty important genes for people's health. But they also found that a lot of the DNA does not code for anything. There's no genes that are used. And what they found over time by mapping out the human genome is that roughly 2% of your DNA is not really is only 2% of your DNA is used to code for genes. It's the only 2% is actually transcribed into RNA. So the real important point of this is that large sections of your DNA is not used at all, and this large sections of DNA can often be come into play and become part of genes later on in the species evolution. So, all right, let's look at sex-linked genes. These are genes that are only found on the two sex chromosomes, the X chromosome and the Y chromosome. And you're going to see when we look at the Punnett square that they're inherited a little differently than the ones on the autosomes. Remember, the autosomes are the other 22 chromosomes. For men, if the allele is found on the X chromosome, because men don't have that other Y chromosome to sort of cancel out what's on the X chromosome, they're going to inherit the traits that are coded for the alleles on the X chromosome. And some of these can include recessive disorders such as color blindness, hemophilia, and muscular dystrophy. And men are more likely to get them because they don't have that X chromosome, the allele on the X chromosome, to cancel out the recessive disease allele that's found on their only X chromosome. And here's a picture of some color blindness tests. And this is actually a fairly common disease. Now, if a woman were to get the recessive alleles, she's going to have two, one on each of the X chromosomes. Let's look at this. Let's look at this here. Here's for color blindness, and you can see we'll represent men as a square, women as a circle. Here's a colorblind man, colorblind female. Here's a normal vision man and a normal vision female. And remember, we have these people that are half coded or carriers. So let's mate a father with normal vision to a carrier mother. And what ends up happening is, if you do the Punnett square out and you distribute the alleles, you can see that there are four possible phenotypes, and one is a daughter with normal vision, one's a son with normal vision because they did not receive the allele on this X chromosome. But if the female receives the allele on the X chromosome, she's going to be a carrier. If the son receives the allele on the X chromosome, he's going to be colorblind because remember, this allele does not exist on the Y chromosome, so it has to be expressed for him. You might ask, can women become colorblind? The answer is yes. It's what's going to end up happening is she's going to have either a carrier mother, and she's going to have the recessive allele here, And let's say a carrier mother marries a father who's colorblind, and this would be in a recessive allele here. So she'd receive a recessive allele here, recessive allele here, so she would be colorblind there. Now, this is an interesting effect for females. Females have 2 X chromosomes, the males only have 1. So to account for this difference, one of the X chromosomes in each of the women's body cells is randomly turned off, and it forms a dense part in the nucleus known as a bar body. And basically, a bar body is a covering over the X chromosome to prevent the alleles, the genes, from being expressed in that one X chromosome. And men don't have these because they have to have their X chromosome active for the alleles that are normally coded for on the X chromosome to be expressed. And you might say, well, why don't women with bar bodies get diseases? Well, 50% of the cells that they have might have a recessive sex-linked disease, but the other 50% of her cells would be normal, and these cells are generally sufficient enough to spare the females from getting the disease. Now, the most common error during meiosis is what's called non-disjunction. And what happens is that the chromosomes can fail to separate properly during meiosis. And what you end up having is gametes with an abnormal number of chromosomes. Sometimes they'll have 3 of one chromosome, sometimes they'll have 1 of one chromosome. And you end up with a different number of chromosome numbers. And one of the examples is what's called Down syndrome, which is when somebody has an extra copy of chromosome 21. Now I have 2 charts here. I'm going to show you this one first. Let's say that meiosis 1 goes as planned, and during meiosis 2, the chromatids normally would split apart from each other. So you'd end up, if the cell normally has 4 chromosomes, the gamete should have 2, and that's what happened here. But let's say this chromosome doesn't line up on the equator and split correctly. Then you might end up with 3 chromosomes in this gamete and less than the 2 chromosomes, missing 1 chromosome in this gamete. Here's what it would look like in meiosis 1. Ordinarily, the chromosomes would line up in homologous pairs and split. But let's just say they don't split correctly. You may end up with an extra chromosome, 2 of these black chromosomes, in this cell right here, and then when those cells split again, now we have 2 gametes that are missing a chromosome here, and 2 gametes here that have an extra chromosome. These are both possible ways that non-disjunction can occur. Now, if you have non-disjunction occur with sex chromosomes, then you can end up with some rather strange disorders that will affect the expression of sex traits in a male or female. In Turner syndrome, a woman may only have 1 X chromosome. She may end up losing the gamete may only have 22. One of her gametes might only have 22 chromosomes, and it would be missing an X chromosome. And in Klinefelter syndrome, a woman could end up with the opposite. She could end up with a gamete that has 2 X chromosomes in it already, and then during fertilization, she ends up with 3 X chromosomes. And you can read through this chart, and you can find these charts online, and you can see a lot of times that the symptoms are kind of what's the word I want? Smaller sex characteristics, and there are it's more than just Turner's and Klinefelter syndrome. This can also happen to the Y chromosome. So that's the end of the 14.2 podcast, and we will talk again soon. Thank you.